Hi, I'm Josh Titlow.
Hi, I'm Zana. I'm going to give a brief introduction about the tension nerve study. Sometimes you might wonder why when you are lifting a heavy object, you drop it very quickly and involuntarily. It happens because the activation of the tension receptors that control and regulate the tension of muscle fibers in order to avoid muscle damage when you are lifting a very heavy object. In the late 19th century, Camillo Golgi found receptors that monitor and sense the strength of muscle tension, which are called Golgi tendon organs. The dendrites of these receptors are located at the end of vertebrate muscle fibers, but they are not directly associated with the muscle fibers. At the beginning, it was believed that the Golgi tendon organs only respond to extreme tension, but later on, it was proved that they respond to low tension too. The research to find a less sophisticated model organism to study tension receptors was continued until the discovery and delineation of tension receptors in arthropods by Macmillan and Dando in 1972. They found out that these receptors are located on the apodemus of the Dungeness crab, Cancer magister flexor muscle fibers at Meares region. Also, tension receptors were found in flexor muscle apodemus of tail spine of horseshoe crab by Eagles and Hartman in 1975. Then Eagle delineated the anatomical and physiological features of tension receptors in tibial flexor muscle apodemus in horseshoe crab. The detailed information of tension receptors was shown by Parson in 1980. He showed that there are about 150 tension receptors in horseshoe crab. Later on, it was revealed that there are tension receptors in jumping leg of locust at femur region. In addition, it was found that there are tension receptors in walking legs of the blue crab Calinectes sapides. Some of these tension receptors are located at the opener muscle apodeme. Then tension receptors are bipolar sensory neurons. The size of the cell body is about 60 micrometer in diameter, and their number ranges from 9 to 15 sensory neurons that are inserted into the opener muscle apodeme. The opener muscle fibers contraction causes these tension receptors to fire action potentials. Then the information might be delayed into the central nervous system that in turn results in the reduction of motor neuron activities. It was shown that neuromodulators such as serotonin increase the tension of muscle fibers. In turn, they increase the firing rate of tension receptors. It was noted that a receptor can be considered as a tension receptor if the following criteria are met. First, the rapid passive stretching of muscle fibers should activate the tension nerve, and also tension receptors usually are not attached to muscle fibers. Then there is no spontaneous activity in tension receptors when the joint is at rest position, and also tension receptors should fire action potentials when isometric tension is applied. But the frequency of firing should increase with increasing of tension. And also tension receptors should fire action potential during tension application, but it should stop firing during rapid release. In order to study tension receptors, we will use blue crab Calinectes sapides. In the following videos, we will show you how to dissect the walking leg to get tension receptors and record their electrical activities during stimulation of opener exciter motor neuron.
Let's have a brief overview of the crustacean tension receptor protocols. In this series of experiments, we will first use methylene blue staining to become familiar with the nervous system anatomy in the walking leg. Then we will record neural responses to passive and active force generation in the tension nerve innervating the opener muscle. And last, we will measure the actual forces with the force transducer. The dissection used for each of the experiments is identical. However, for the physiology, we only lightly stain the preparation to minimize cell destruction. In the crab walking leg, the opener motor nerve is exposed by removing a small section of cuticle in the carpus or in the meris. With methylene blue, the idea is to visualize the anatomy so later the dissection can be formed with only light staining or no staining at all. The results should provide a vivid contrast between the neurons and muscle tissue to give a beautiful layout of the sensory and motor nerves in the walking leg. Feel free to bring in cameras and capture photos to commemorate the dissection. The first physiological experiment involves recording from the nerve with the opener joint positioned at various angles. The experiment demonstrates the neural activity associated with passive tensile force on the opener muscle provided by the joint rotation. In the second physiological experiment, we'll stimulate motor neurons directly to develop tension in the opener muscle. Be careful not to overstimulate the nerve, as this will cause synaptic fatigue. The goal with these extracellular recordings is to characterize neural activity in the tension nerve and relate it to the active and passive muscle tension. In the last experiment, we will repeat the joint angle series and record the force generated with a force transducer. The goal here will be to quantify the forces achieved by rotating the leg or activating motor neurons. Ideally, we would record the force while recording from the tension nerve, but the force transducer produces 60-cycle noise that masks the recordings. We encourage you to try both recordings simultaneously, but note that this approach can be used if the signal is too noisy. As you perform the experiments, think about how the architecture of the sensory organs enables the nervous system to detect tensions in the muscle tendons. Also think about how this information is transduced or coded in the neural circuitry and how the central nervous system perceives this information.
Then I will catch one of the crabs to take one of the walking legs off because you need it. Then this one seems to be very active, and you can do it like this. If you look at the kilopedias, you can see that there is a rubber band on it. This is for protection because they will pinch you if there is no rubber bands on it. Then we will use the middle nose pliers to pinch one of the walking legs here in this region. You don't have to pinch it here. When you pinch it here, then you see that it will donate the leg. Pinch it gently. Then you can see the site of autotomizing here. And we have a nice walking leg to dissect it. Then now I will put it back into aquarium here. Then we can use the other walking legs next time. Then I'm going to dissect the walking leg that we just took off from the crab. Then I'll show you how to dissect this preparation and how to use methylene blue to stain the prep, because at the beginning we have to use methylene blue to see all the nerves and muscle fibers, because the tension nerve that you are looking for in this lab is very small. If you don't stain it at the beginning with methylene blue, it will be very difficult to find it later on when you do electrophysiological recording. But here, when you use methylene, you don't have to worry about the electrophysiology because we will dissect another leg to do electrophysiology. Then this leg, if you look at the anatomy of this leg, like the gross anatomy, then you see we will dissect this part. It's called carpopodi, and this is the right walking leg of the crab. Then here there is a type of muscle called bender muscle, and this is a medial side called, and the outer side called lateral side. There is a stretcher muscle. When this muscle contracts, then this limbs moves like this. Then the motor neuron that we will stimulate it when you do electrophysiological recording is located underneath the bender muscle. Therefore, we have to make a window here and remove the cuticle, then remove the bender muscle. Then after that, we will dissect the propodite, and we also, if you look at this region, this region is dorsal region, and this is ventral region, and the opener muscle is located here. with a tension nerve, and the closer muscle is located in ventral region. We don't need the closer muscle. We should remove the closer muscle in order to see the tension nerve. Then let's do the dissection now. Let's begin the dissection. Then at the beginning I use the blade with this scalpel with blade number 11. At the beginning I will dissect the carpopodite region here to make a window. Then to get a motor neuron, then I will dissect the propodite region later on. Then here you should be careful when you cut it with the blade. You don't have to go too deep because you will cut the nerves, then you don't get any activities. Be sure that you cut the cuticle all the way. And you can cut the muscle like this underneath. You see that you can remove the cuticle very easily. Then, as I mentioned before, this part is a bender muscle. Then we don't need the bender muscle. You see that this is this white structure is a tendon. We should cut the tendon here with the blade number 11 and cut it like this. And after that you can remove it like this. And now we are done with this part. Then we should put inside the saline before it dries out. And we have craft saline. Then we can make this simple tool. This is a very simple tool. You can make it in the lab. And this is a insect pin. And with this stick, you can like make a hole here with a big pin. Then use this small insect pin. Then now we are going to try to find the motor nerve here. Then we should find the motor nerve. Then like I said, we will use but you can try first to see if you can find it, but later on we will stain it with methylene blue. Then you can like know the location of the motor neuron here. Then later on you can find it easily without staining. But you can use this simple tool to find the motor nerve. I can see it now here. There's a big nerve bundle. Then on this side, look at on this side, then you can see here a big nerve bundle. A branch of motor nerves comes out here and goes to those muscle fibers. Then we can stimulate this motor nerve. It's called antidromically stimulation. And then when you stimulate it, it goes back to the main motor nerve and goes down again, which stimulate the opener muscle fibers. Then now I'm going to dissect the propodite segment. Like I said, the opener muscle is located at the dorsal region and the closer muscle is located at the ventral region. Then the tension nerve is located close to the opener muscle. Then we have to make a window here like this, then remove the cuticle, then after that remove the closer muscle. Then we can see the tension nerve very easily. Now we should remove the cuticle. Then remove the connective tissues here. Then you can see that this is a tendon with those muscle fibers that are closer muscles. Then we can cut the tendon here and remove it. And the nerve bundle is located in this region here. Then now let's cut the tendon because we don't need it. And we can pull it back like this. And remove it. Also, you can see this is a nerve bundle here. And also we don't need this nerve bundle because the tension nerve is located on, it's close from the motor neurons. If you remove this nerve bundle, it doesn't matter. Now I'm going to remove the nerve bundle, but it's better to separate the nerve bundle like this with this small insect pin, then to see the boundaries very well. Then you'll see that there are a lot of blood vessels there. Then we can use a blade to cut the nerve bundle at the end. Then we can remove the nerve bundle. Now it's ready to stain it. Now I'll pour the saline into the saline beaker. Then I will use a cold methylene blue to stain the nerve fibers here. Then I will put inside the refrigerator for 10 minutes because a cold methylene blue is good and it stains the nerve fibers very well, and we can do this many times before put inside the refrigerator. Then if there's something here, we can remove it. This is the tendon and muscle fibers. Now it's ready to put inside the refrigerator for 10 minutes.